to welcome you all to the first session of the year of Cerrahpasa Neuroscience Talks. Today, we have the great privilege of hosting an exceptional scholar and a leading expert in the neuroscience of free will, Dr. Aaron Schurger. Our focus today will be on a fundamental topic regarding how our brains controls movements. We will explore neural antecedents of spontaneous voluntary movements. As we wait for the talk by Dr. Schurger, let us first get to know his impressive background and accomplishments. Dr. Aaron Schurger is currently an assistant professor in the Department of Psychology and the Institute for Interdisciplinary Brain and Behavioral Sciences at Chapman University. He received his bachelor's degree in computer science from Indiana University and obtained his MA and PhD degrees in psychology and neuroscience from Princeton University. Before joining Chapman University, Dr. Schurger held research positions at leading European centers, such as the National Institute of Health and Medical Research in France and the Civil Federal Institute of Technology Lausanne in Switzerland. He is best known for his pioneering work on the readiness potential. This work challenged the classical interpretation of the Libet experiments with the stochastic accumulation of noise model. Dr. Schurger is also the recipient of prestigious awards, including the William James Prize and the ERC Starting Grant. It's a great privilege to have Dr. Schurger as our guest. Today, he will guide us through the history of research on this topic, going back to the discovery of the readiness potential in 1965. He will discuss how the neural commitment to initiate movement actually happens, much closer to the moment of action than previously thought. Crucially, Dr. Schurger's work underscores a general caveat about time series data, that what seems to be a very reliable antecedent is not necessarily a good predictor always. We also look forward to the insights he will share on how best to carry out this kind of research in order to avoid the pitfalls associated with such analysis. After his presentation, we will have a Q&A session. So without any further delay, I welcome Dr. Aaron Schurger. I'm looking forward to his presentation on neural antecedents of spontaneous voluntary movements. Professor Luck, welcome again. The floor is yours.
Okay. Wow. Thank you very much for that very nice introduction, and an excellent introduction, I might add. You hit all the important points, and you got it all right.
Nice to hear that.
So thank you. No, this is a great pleasure to do this, and an honor to be able to share some of this work with you. In 45 minutes, I won't have time to share all of it, but I've tried to share some of the key parts and some of what we're doing more recently. So let me share my screen, first of all, before I forget. Sometimes I start talking and I don't even share my screen. So let me remember to do that. Okay. And I don't have any sound, so I don't have to worry about that. And then
We can see that.
great. Okay, wonderful. So I like this image. I share this image of a cloud and of rain because it helps me at least to think of behavior and the system that's generating the behavior in a more abstract and scientific and detached way. So when we think of a decision, we often immediately think of a conscious decision of, my decision that I will go to the store after work today, things like that. But there's of course another sense of a decision, which is what are the neural events in the brain that cause the action in the muscle and in the body. Those might be linked, and in fact, I believe that they are very tightly linked, but still they are not the same thing, and we know that from many years of research. And so you can kind of think of a rain cloud as a complex system that has at least one behavior, which is dropping rain. And at some moment, this behavior starts. And you can think about trying to ask the question, well, what are the antecedents of that event happening in that system, right? And how well do those antecedents predict that particular behavior? So in the case of the human brain, the human body, um, you can think of this kind of decision uh, as what I call a spontaneous voluntary action. And of course, when I say spontaneous, I mean spontaneous from the third person perspective. And a good example is the diver who is on the diving board and is going to dive, is going to perform a dive. Um, there isn't any stimulus there in that context. There isn't any immediate stimulus that tells the diver exactly when to dive. They reach the end of the board, and then ultimately, precisely when they initiate the movement, when they initiate the dive, is up to them in some sense. There's no bell, there's no visual flash, there's no other kind of stimulus that says dive now. So they have to, when for some reason, presumably when they are ready, whatever that means, they initiate the dive. Um, and there's a question there. There are many questions there, but there's a very fundamental one, which is why did they initiate that action precisely when they did, and not, for example, 1 second earlier or 1 second later, right? Or 500 milliseconds earlier or 500 milliseconds later. Why did that happen precisely when it did? And that's a very essential part of the kinds of questions that I'm asking in my lab and in my research. So what is happening in the brain when you initiate an action spontaneously, like in this context? And how well do those events in the brain predict that the event is going to happen? Just like we might try to predict when the cloud is going to drop rain. I often think of this, and this is a quote um, uh, from a book called Atonement. Um, I think of this as the dividing moment between not moving and moving. Um, and I often call it the neural decision to move in order to distinguish it from the conscious decision to move. And it's the hypothetical neural event that commits some part of the body to an imminent movement. In a sense, like knocking over a domino in a row of dominoes like this. So it's a commitment in a sense, but it's not deterministic. Meaning, even in the case of dominoes, if I knock over the row of dominoes, I could at some point intervene and prevent it from reaching the end, right? But if you leave it alone, all other things being equal, it will reach the end. So in that sense, it's a commitment to movement. And this is the way that philosophers think about an intention. So according to most philosophers, an intention is a commitment to a certain course of action or specific action. Now, in neuroscience, the very earliest work uh, we know of, specifically looking at this kind of context of spontaneous voluntary movement, goes back to the year 1965, when Kornhuber and Deka um, uh, produced their paper reporting their discovery of the cortical readiness potential. And of course, this was originally published in German. Um, they were, of course, two German neuroscientists. Um, translated into English, this is changes in brain potentials with willful and passive movement in humans, the readiness potential and reafferent potentials. In German, this is called the Bereitschaftspotential, but in English, we translate it as the readiness potential. And the readiness potential, some of you may be familiar with it, is a slow buildup of electrical potential over the motor cortex recorded using scalp EEG. Here you see it recorded from two sites, two precentral sites, right and left, roughly corresponding to right and left motor cortex. And you see this is a slow building of electrical potential that precedes, here's time zero right here. This is the time of the movement itself. It precedes the initiation of the movement by up to one full second or even more. And notice here, this is negative electrical potential, as was the custom at the time. Negative was presented up. You'll see in other plots, negative is going down, which is now the custom more recently. What was unique at the time about Kornhuber and Dicke's experiment was that there was no stimulus, there was no cue, right? So this was not a stimulus response paradigm. It was just a response. Subjects were asked to perform self-paced or uncued movements. And they were asked, don't tap a rhythm with your hand, but just make a movement when you want to, every so often at irregular intervals. And in their experiment, I believe the movement was a simple flick of the wrist. Now at the time, they didn't have computers. They didn't have much of the kind of equipment we have today. So they had to record these electrical potentials on these reel-to-reel tapes, which you see in the background here. And when the movement would happen, then they would stop the tape, take them off and reverse them and play it back in reverse. That was how they managed to get this kind of data, just a back average. So since its discovery, the readiness potential has been interpreted as the electrophysiological sign of planning, preparation, and initiation of volitional acts. That's been the classic and the dominant prevailing interpretation of this. And you see this not just in EEG, by the way. You can also see this same sort of buildup if you record from single neurons in areas of the human motor system. So these data here are from invasive electrodes placed inside the brain of epilepsy patients. These data come from the pre-supplementary motor area. And here, time zero is the time of the movement. And you see, again, this buildup, in this case, in firing rate that precedes the movement by more than a full second. And this same kind of buildup, which we can call the pre-movement buildup for short, is also been found in primates, in rodents, and even, oddly enough, in crayfish. And that's interesting because crayfish are invertebrates. They don't have a motor cortex, and in fact, they don't have any cortex at all. So this came from another part of the brain of the crayfish, a motor control structure in their brain, but not their cortex, which they don't have. But it's interesting that this same phenomenon appears very broadly across the animal kingdom. Some years later, so almost 20 years later, Benjamin Libet published his now very famous study on the time of the conscious intention to act in relation to the time of the readiness potential. So he knew about the readiness potential. It by then was a very well-known finding. His addition to the story was to ask when in the time course of the readiness potential, the time leading up to the initiation of a movement, when do you feel your conscious decision or your conscious urge to perform that action? So in order to do that, Libet recorded EEG over the motor areas, recorded the readiness potential, but at the same time, he asked his participants to monitor a clock. The clock was a rotating dot going around a dial, and the time of this clock, the time scale of this clock was not like a usual clock. It made one full rotation in 3 seconds. So the dot would make its way around the clock face in 3 seconds, which meant that each second on the clock actually corresponds to about 50 milliseconds. And Libet asked his subjects just to perform a quick, again, abrupt movement, which was a flick of the wrist. Said let the urge to act appear on its own at any time without pre-planning or concentrating on when to act. So he essentially asked his participants to try to be spontaneous, which is a strange instruction, by the way. That is definitely a strange kind of a task. Interestingly, people can do it. So if you ask your subjects to do that, they may find it strange at first, but after trying a few times, they kind of find some way to do that. They find some way to generate what may seem like random movements. But so Libet's big question was, if you look at the time course of the readiness potential here, leading up to 0, and you ask people to monitor the clock, and you ask them to report back, what time was it? Where was the dot on the clock when you first felt that urge to initiate the movement? Now, the famous outcome of his experiment is, of course, at time 0, you move. Sometime well in advance of that, where the readiness potential appears to begin, the brain is presumably getting ready to move. But the interesting finding was that the conscious decision actually happened much closer in time to the movement itself than the readiness potential, than the onset of the readiness potential. And of course, this had important implications philosophically and just more generally with theoretically around the idea of freely and consciously initiating movements, right? The idea being that if your conscious decision happens after the brain is already getting ready to move, then how can that be an act of conscious free will? It would seem that it can't. I won't get into that more philosophical debate, but rather what I'm going to talk about is how we can interpret this kind of data in a different way. So Libet's experiment has really led to this prevailing view where very early in time, maybe 1 second or even more before the movement, there is a decision. There's this neuronal commitment to initiate the action. And all the rest of this unfolding in time, so the readiness potential itself, is the outcome of a decision, of a neural decision, right? It's planning and preparation for movement. And then very late, or relatively late, in that sequence of events, we have the time when the self becomes aware of the conscious decision to move. Right? So this is the prevailing view that I've kind of caricatured here with this picture of dominoes. Now, this prevailing view leaves us with some very puzzling questions. And those questions have been there ever since this view was proposed in 1983. So I have a list. I list here just a few of those puzzling questions. One is that humans are quite accurate in estimating the time of sensory perceptual decisions. Libet himself tested that. He used his clock to estimate how precise people could report the a touch stimulus to the skin. And later work has looked at how well it can estimate, how well subjects can estimate the time of an auditory beep or a visual flash. They're quite good. They can estimate the time of those events with an accuracy of plus or minus roughly 50 milliseconds. So then that begs the question, well, why not motor decisions as well? Why are they hundreds of milliseconds behind in estimating the time of their own motor decisions? That is, when you ask them to report when they felt the urge or the decision to move. Another puzzling question is why the readiness potential has not proven to be a very informative signal feature for brain-computer interfaces. So many labs, many scientists and engineers have tried to use the readiness potential to predict the onset of movement, use that to create an asynchronous brain-computer interface. It just doesn't work very well. Right? There have been some published attempts. There have been many unpublished attempts that simply failed. Among the published attempts, right, the accuracy of the RP as a signal feature for predicting upcoming movements is never very good. Never better than about 60 or 70% correct. And then finally, another puzzling question is why does the crayfish have a readiness potential? Right, even though it has no motor cortex or even no cortex at all. Right, this would seem not to be a specifically human or even mammalian phenomenon, or even a cortical phenomenon, but seems to be something much more primitive, much more fundamental. And it's unclear how Libet's view would account for that, or any of these questions. So what I've done in my work, and this started really back in around 2010 now, was try to offer a new perspective on the readiness potential and on what it means. And so I'll summarize that perspective here in a few points. One is that there is a threshold in the motor system for triggering a movement. And when neural activity in the motor cortex reaches the threshold, then you move. And of course, that's specific to the particular kind of movement, to the particular movement, whether that's a hand movement or a foot movement or whatever. And when the imperative to move is relatively weak, then the precise time of the threshold crossing is partly determined by noise in the motor system. Now, what I mean by the imperative to move, right, is the difference between a task like Libet's, where you're asked to kind of move whenever you want to, right, versus a task where you have, for example, a precise stimulus like an auditory beep, and I say press the button when you hear the beep. So in the latter case, the imperative to move is strong. If I tell you, wait for the auditory sound, wait for the beep. When you hear the beep, press the movement. That beep is an imperative. It says move now. So that I would consider to be a strong imperative. In Libet's task, there essentially is no real imperative or very little, right? Just move whenever you want to. In that context, right, it might be possible that random fluctuations, what you might call noise in the motor system, might partly determine the exact time of the threshold crossing and as such, the exact time that you initiate the movement, right? So as a consequence, then, the time of movement onset will tend to align with crests in the noise. And that's what gives us the readiness potential. And so this is essentially this perspective that I've proposed on the readiness potential. And I'll go through this a little bit more carefully and look at some of the evidence for it. So the first thing that we have to take account of is, well, do we have spontaneous fluctuations in neural activity in the motor system? Do we have this kind of noise? And we do, absolutely. So even if you look at resting EEG data or resting intracranial data for that matter, you find that the system is never completely silent, right? There are ongoing random fluctuations. And notice that those ongoing fluctuations, that background activity, which you can think of as noise, right? It's not white noise, which is what I have shown at the bottom here. If you look at resting EEG data or resting intracranial data, you don't see this kind of noise, which again is white noise. If you see white noise, pure white noise, then there's either something very wrong with your equipment or your experiment participant is literally dead. Um, what you do see is this kind of noise, what I've shown up above, autocorrelated noise. And to to explain simply what autocorrelated noise is and how it's different from white noise, first of all, it's often called pink noise. This is a kind of noise that's dominated more by the slower fluctuations. And the faster, higher frequency fluctuations are less present in autocorrelated noise. And you can see that here just by looking at it. You see that this drift, this slow drift, is responsible for these larger deflections. And the faster activity here, it's there, but it's smaller in amplitude. Right? So this kind of noise is dominated by slow drift. And this is the kind of noise that you see in general in nature, and not just in neural systems, but definitely you do see them in neural systems across all scales, from microscopic scales to variance in reaction times, for example, at the behavioral level. So one way then to think about this account is to ask a question: how might a machine comply with Libet's instructions? So supposing you wanted to build a simple machine, for example, a robotic arm that could comply with Libet's instructions. So it will perform a movement at random times, right? Random and let's say even unpredictable, even to itself. And one possible solution to that question would be to simply bias or move the baseline of your activity closer to the threshold or lower the threshold, either way, same thing. And then just wait. And if you're relatively close to the threshold and you just wait, eventually you'll cross the threshold. This system will cross the threshold at a random time, right? Just randomly, right? And if this is the threshold for your actuator for causing the movement of the robotic arm, then at this moment the robotic arm will move, right? And the precise time of those movements will be more or less unpredictable, would be more or less random, even to the system itself. So this may not be the only solution to that problem, to that question, but it's a solution that can work. And we wonder, and I wondered, if maybe this isn't part of the way that the human brain complies with Libet's instructions, right? Doesn't have to account for everything, but it might be part of the solution. So in order to test this idea, I worked with what I call the stochastic decision model, which is nothing more than an accumulation to bound model that's commonly used to model perceptual decision-making tasks, right? So you have in this model, which is a simple differential equation, you have a drift term, and the word drift comes from the drift diffusion model in physics, which is what this is based on. That's the signal, or what I call the imperative, right? And in this context, like I said, the imperative is very small. There isn't much of an imperative in this task, but it's not really zero, and I'll explain why in a minute. You have a leak term. This is a leaky stochastic accumulator. So this leak term turns into exponential decay in x, right? And it just simply means that the system has a tendency to decay back to zero if you remove any input. So if you remove the imperative, this will eventually tend to decay back to zero. And then this being a stochastic process, you have Gaussian noise here. Now, one thing to note, that if you take the integral over Gaussian noise, you end up with autocorrelated noise. And that's how this kind of system produces these time series that fluctuate with significant autocorrelation. So it produces pink noise. right? And there's of course another part of this which isn't shown here, which is there's a threshold on X. So somewhere you set a threshold so that when X exceeds that threshold, something happens, right? Something is triggered. So I use this model to generate these kind of time series. So you start the model. Um, be, what happens with a leaky stochastic accumulator like this is that the signal tends to climb a bit, and then it tends on average to level off at a point given by I over K. So where the imperative and the leak kind of balance each other out. So it becomes stationary at around that point. So that you can think of that roughly as moving closer to the threshold and then simply fluctuating randomly around that point. And if you have an appropriately placed threshold, then eventually this signal will cross. And um, that crossing time, and that's given by these upside down arrows, that crossing time can happen earlier or can happen later. Um, and I'll show you what the distribution looks like. But then you can use this to model the task where you say, well, when this thing crosses the threshold, that's when the movement is initiated. And so what do we do in an EEG experiment or any kind of experiment when we're looking at the readiness potential is we take the data, we find out when the movement happened. We align the data to the time of the movement, and then we take the average. So I did the same thing with the model. I took the first crossing time for each instance of the model. I aligned the data to that time, uh, and then took the average. And in order to be able to compare it to the readiness potential, I reversed the sign. And indeed, when I did that, I found something that has something that very much resembled the shape of the readiness potential, this gradual, slow, nonlinear buildup. So once I discovered that I could approximate the shape of the readiness potential, then I used this model to fit real data. So I had subjects come in and do Libet's task. I recorded their empirical readiness potential, and I did some model fitting to see how well I could fit it. I began with the waiting time distribution, which you see on the left. That's the distribution of how long participants wait before they randomly and spontaneously make their movement. And that's entirely up to them. They wait for a little while and then they move. If you look at the distribution of those waiting times, I found that I could approximate it very nicely with the distribution of first crossing times from the model. And using those very same parameters, then I fit the resulting trajectory of the decision variable to the shape of the readiness potential, right? Up to about 150 milliseconds before zero, which is how long it takes for activity to make it from the motor cortex to the muscle and to start initiating a movement. As you can see here, the fit was very nice. So the model fits the data very nicely, and so can account for the kind of data you get in Libet's task, right? One question is, well, why is this imperative term here very small? Why is it not zero, right? The reason it's not zero, the reason I chose to have a small imperative is because in this kind of a task, there actually is an imperative that's given by the demand characteristics of the task, right? So when a participant comes in to do this experiment, you do tell them in the instructions, like you can move, whenever you want. It's just up to you. There's no stimulus and you just kind of wait. And whenever you feel like it, you can move. That is, again, a strange instruction, but technically speaking, they could wait 1 minute, right? If they could wait 5 minutes if they wanted to, They could wait till the end of the experiment. They could wait an hour. They could never move, right? Those are all permissible. But in actual fact, nobody ever in the time I've been doing this experiment, nobody ever waits longer than about 30 seconds at the very longest. Typically, they're more around like 7 seconds is the typical waiting time. Why is that? Well, that's the demand characteristics of the task. The participant knows, even if I don't tell them, they know that this is an experiment about making movements. And they know that I'm probably, even though I didn't say it, I'm probably expecting them to move because that's my data. I need those. And so wanting to please the scientist running the study, they move, right? And they don't wait too long to do so. So there is an imperative in this task. It's just a very weak, nondescript kind of imperative. So that's why that value is not quite zero. And that is an actual important parameter in making the model fit the data well. So when you have a model that seems to fit the data well, one good way to test it is to come up with a novel prediction. So we did. This model offered a prediction, and I'll explain what that prediction is in a moment. But what we did in order to test that prediction, we had subjects do the standard limit task, and after they were done, we had them repeat that same task again. But this time we told them you're going to hear an occasional click, an auditory click that you might hear. Maybe you won't hear it, but you might hear a click at a random time during the trial. If you hear the click, you should, in this case, subjects were pressing a button. If you hear the click, you should press the button immediately, as quickly as fast as you possibly can in reaction to the click. Otherwise, if you don't hear the click, it's the same task. Just wait for a while, and whenever you feel like it, just press the button. So this context, right, actually offers us an interesting kind of prediction. And here I'll describe the prediction. So here we have our threshold, which is the dashed line, and we have our kind of randomly varying autocorrelated signal, which is near the threshold. Okay. All other things being equal, if there hadn't been any particular task here, movement would have happened here, right? The first time that the signal actually crosses the threshold. It would have. So remember, we tell them that we might interrupt them with a click. Well, look at what happens if the click had happened at this point in time, right here. Okay? If it had happened here, and the outcome of the click producing a movement, imagine that requires taking the state of your motor system and bringing it to the threshold as quickly as possible. That's how we modeled that. So if the click had happened here, well, at this point in time, just for random reasons, the signal is a little bit farther from the threshold, so it would take a little bit longer for the signal to quickly ramp up to the threshold, and that will give you your reaction time to the click. On the other hand, if the interruption or the click were to happen here, then that resulting reaction time would be that much shorter because the motor system is already pretty close to the threshold as it happens. So you'd have a shorter reaction time. Then, of course, if the click were to happen out here, well, then it would just simply, if it was programmed to happen out here, it would just never happen because the subject would have already made their spontaneous movement, right? Now, the moment at which these interruptions happened was random. It was unknown to the subjects. It was unknown even to me. So the prediction then is this, at what we have at the bottom, is that the fast reaction times to those interruptions should be preceded by a buildup in signal amplitude beginning well before the interruption itself. right? Even when the subject was not preparing to move at that particular moment. And that is indeed what we found. So in the model, and you can look here on the left, is the data time locked to the movement. And on the right here are the data time locked to the interruption, to the click. Okay? These are just two different points of view on the same data. When you look at the model and you see here, what we've done is separated the subject's relatively slower responses to the click and their fast responses to the click. And you can see in the model that the fast responses to the click, here is 0. This is the time of the movement. Over here, 0 is the time of the click. So you can see in either case, for those fast responses, you have some kind of slow ramping already happening before the event in question in both cases. And we find the same general pattern in the actual EEG data, which we have above here, right? So you see when time locked to the movement, their fast responses are preceded by a slow, gradual buildup, beginning well before time 0. And this is true even when it's time locked to the click. So here's the click itself, and you see there's already a slow buildup that's significantly different from when their responses were slow. And really the important thing here is that this buildup cannot be a preparatory signal, because you cannot prepare for a movement that you don't know you're going to make, right? They didn't know they were going to move at time 0 because this was a random interruption. So that suggests then that the reason for this difference is because of what we showed in the model, that they were fast on the fast trials precisely because just by chance their motor system happened to be that much closer to the threshold already. Okay, so we've done some other experiments and other tests that I won't get into, but I'll summarize with this being our now different view of the readiness potential, which is simpler in a sense, that this early part of the readiness potential, going back 500 milliseconds or more, sometimes 1 second, 1.5 seconds even, that's accounted for by slow, spontaneous, random fluctuations in the motor system. And then what happens starting at about 150 milliseconds before the movement, that is the actual neural decision to move. That's the actual commitment, right? And we argue that that is a threshold crossing event and happens at about 150 milliseconds before movement. So if we go back, if we take this view and go back and look at our puzzling questions, what's nice about this view is that it answers those questions very nicely. So for example, remember, subjects are accurate in estimating the time of their sensory perceptual decisions, right? Why not their motor decisions as well? And the simple answer there is, yeah, why not, right? Maybe when Libet's subjects and when subjects in general who do this experiment, when they report feeling the urge to move at about 200 or 150 milliseconds before the movement, maybe that's because that's when the decision happened. Maybe Libet's mistake was to not take his participants at their word, right? They said, hey, I decided this is when I felt the decision happened. Libet assumed, oh, well, they must be wrong because the readiness potential goes back much earlier than that, right? So the answer might be, well, yeah, maybe that's precisely when the decision happens, and that when you're asked to report consciously when it happens, they line up. They happen at about the same time. Remember this other puzzling question: why hasn't the readiness potential proven to be a very informative signal feature for asynchronous brain-computer interfaces? And the reason there is simple, is that if it's governed in part by random noise, then very often, or at least sometimes, the signal might come quite close to the threshold, but not cross it, right? And it'll be then hard, very hard, to develop an interface based on that signal alone, um, that has any high degree of sensitivity. And one thing that this suggests is that the limiting factor for these kind of brain-computer interfaces would be the false alarm rate, the false positive rate. And that is, in fact, what happens with scientists who are working with brain-computer interfaces. That is almost always the limiting factor, including when those are driven by intracranial electrodes, right, with a very high signal. And the final puzzling question, the crayfish, remember, also has a readiness potential with about the same time constant as the human, but has no motor cortex or even any cortex at all. Well, under this view of the readiness potential, all that's necessary for that to be true is that the time constant for these kind of background fluctuations in neural systems in general has been relatively conserved across evolution. And that turns out to be true. If you look, for example, at an animal as simple as a leech, the time constant for their autocorrelation is very similar to what it is for a human. So that means that if you use the same kind of explanation for why they initiate movements when they do, why they initiate transitions from resting to foraging at a certain moment in time, the time constant for that buildup would be likely to be similar to what it is in a human. And that is, in fact, the case. So this is our roughly new view, the stochastic view of the readiness potential. It's a late decision account in the sense that it argues that the actual real decision the real commitment to initiate the movement happens late at around 150 milliseconds before the movement. And since we first published this account in 2012, there have been a number of other papers that have supported that view empirically in different ways. Here are some of them here. So there are other elements to this history. So I have here Libet's paper. Here is Fried et al., which did the same thing as the Libet task, but with intracranial electrodes. I showed you that data. But there have also been some studies in fMRI, notably coming from John Dylan Haynes' lab, also showing that you can predict better than chance what decisions people will make several seconds later. Some of you may be familiar with these studies. One of the upshot of this studies, one of the important facts and details about these kind of studies is that the predictive capability, so in these studies, you take the fMRI data, you use a machine learning classifier to try and predict, based on the brain data, what choice a person will make some seconds later. If you notice here, the accuracy of these predictions is barely 60%, where 50% is a random guess. Right? These are binary decisions, so you're choosing between two options. So yes, a classifier can predict these better than a random guess, better than 50%, but not very well, right? 58, 59, 60% correct, right? The question is, does that count as a commitment, right? Or is that consistent with simply background conditions that correspond to a tendency, and a slight tendency at that, to act one way or the other? And I think that's in an experiment like this, that's almost to be expected, right? It would be strange not to be able to predict a little bit well in advance, right? As if, for example, those decisions simply came out of nowhere, and they don't. And here I have a quote from John Dylan Haynes himself, says that if you believe that the brain is governed by the laws of nature, that our decisions are realized by our brain, then our decisions should not be completely unrelated to brain activity ongoing in the recent past. So basically agreeing that this may not really count as a commitment. So that's an important caveat here, that above chance classification, if you're trying to predict in the future, is not by itself sufficient to infer a decision or a commitment. Modestly, even significantly above chance accuracy is consistent with background conditions that might bias, but do not fully determine the decision outcome. Okay, so with that caveat in mind, we set out to map the time course of cortical activity that's predictive of impending movement, trying to distinguish the early versus late decision account of movement initiation, right? And what we're looking at here in this study is a fact that in a normal scientific experiment, where, for example, you wanted to isolate what happens before a movement, you'd want to have some data with a movement, right? And you'd want to have some data without a movement, and you'd want to compare those. The problem, though, in the study of self-initiated movement is that you typically don't have any of these. You don't have any epochs without movement. And the reason for that is that you use the movement itself as the signal that you align your data to. So in order to find out where do I look in my data for something interesting, well, I look at where the movement happened, right? So I take every instance where there was a movement, I align the data to that time, and I take the average, right? That's all. I have data when there was a movement, but I don't have any data when there was no movement. One way that people have gotten around that is to take an early period of time, what they call a baseline, and use that as the quote unquote no movement examples, right? You can do that, but the problem is that that's biased because your baseline in that case always has a fixed temporal relationship with the onset of a movement. So it still doesn't really satisfy the criterion here that you need. So you can think of that, and I've talked to weather forecasters about this in this way, that if you have only data that end or that include this event, where for example in weather forecasting the event might be the onset of rainfall, then you will definitely be above chance, what you call chance, in predicting it if you just take advantage of autocorrelation in the data. So one example is if you ask a child, what's the weather going to be tomorrow? If they just say that the weather tomorrow, they say, oh, well, it's going to be the same as today, right? If they say that, then they will in fact be correct much more often than not. They'll be above chance. But notice that doesn't require that they know anything in particular about tomorrow's weather, right? Other than the simple fact that weather patterns tend to be autocorrelated across time, right? So a model like this one, where prediction accuracy gets higher and higher, and this is on random data, right? With random data just taking advantage of autocorrelation, it would seem that very early on you can tell what's going to happen next, right? But in fact, this is just because of autocorrelation. This is the model that you have to beat, right? This is the null model, not 50% correct, which is the mistaken assumption. So in order to have a true control condition, we designed an experiment, and this is work that's been headed up by my PhD student, Luca J. Bizot, and that we did in collaboration with Rob Shapire, who at the time was at the Department of Computer Science at Princeton. He's now working for Microsoft Research. and his PhD student, Mehmet Basbug, who is now working for a startup. And these experiments were done with both MEG and EEG data. So what we did was we had subjects watch a slideshow with pretty pictures. These are just photographs. They're neutral photographs that are nice to look at. It's just a slideshow. Okay. And they can either press a button to advance to the next slide whenever they want, right? Or on some trials, they don't press a button. They just wait, and it advances by itself after a little bit of time. And we label these as automatic or manual trials, and they know which kind it is because there's a cue right before the photo shows up. Importantly, the viewing time on these timed viewing trials, on these automatic trials, the viewing time is drawn from the subject's own viewing time distribution on manual trials, so that by the end of the experiment, you have these two conditions that are roughly matched for waiting time, for how much time elapses, and that end at the end of that episode, they end with a slide transition. Okay? And of course, roughly half the time, that slide transition is accompanied by a movement, and roughly half the time, it's not accompanied by a movement. So now we have these two kinds of trials. We have a movement condition, where you have the movement plus the slide transition happening at time 0. And you have this matched control to condition, where you just have the slide transition, and you waited just as long on average with the same kind of expectancy and anticipation. So what we do with these data, so we have automatic exemplars, manual exemplars, is we perform some machine learning in a sliding window, and we want to look at our classification accuracy as a function of time leading up to time 0, where we're trying to classify, is this a manual episode or is this a automatic one? Right? And of course, we align all of our data to the leading edge of the window, meaning that the future is off limits. So the classifier cannot see anything into the future. They can look up to the edge of the window, and that's it. When we do this, and we've now recorded many more subjects. These were our initial subjects that we ran. These 3 across the top, these are, they came back for 4 sessions each, so we got a large amount of data for them. These down here only came in for 1 session. What you can see is that our ability to tell those 2 kinds of epochs apart is more or less flat at around chance until about 100 or 150 milliseconds before time 0, before the movement, at which point then classification rises rapidly and gets quite high. And the blue line here is MEG data, the yellow is with EEG data. These were recorded simultaneously. So looking at these data, it looks like the decision, right, the information about actually initiating the movement is coming very late. If we just average the data in the old-fashioned way, we find the readiness potential, which goes back a second or more in time across these subjects. What happens if we do this analysis without looking at the no movement trials? Supposing we do this in the way that the field has done it all along, we just have the movement trials, we compare to an early baseline, right? And we ask the machine learning algorithm to tell us whether this is a test epoch or whether it belongs to the baseline. When we do that, right, and that's the blue lines here, when we do that, it looks like we can classify very well, very early, right, compared to the yellow lines, which are the classification with the control condition, right? But this early ability to classify here, this is an illusion, right? It's false. It's, again, based on taking advantage of autocorrelation in the data. Um, the yellow line is what you actually get when you have a proper control condition. Okay, so it looks as if the true decision to initiate the movement happens late, right? Happens maybe around 150 milliseconds before the movement. And why 150 milliseconds? Is this a magic number? Well, it does correspond to some interesting data elsewhere. So, for example, it's associated with an abrupt increase in corticospinal excitability. It corresponds nicely with the subjective estimate of the decision time, around 150 to 200 milliseconds. You see it in feature importance in ECOG in the monkey, that those features become important for detecting movement at about 150 milliseconds before. And it also corresponds nicely to the estimated point of no return in data. So this is the point at which a subject can no longer withhold movement that they're preparing to make. Just a couple of last points. So, some people have asked, well, if 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 this is about noise in the motor system, then why don't we just make lots of random movements all the time? And of course, the answer to that is that this explanation is meant to account for situations where the imperative to move is very weak, where you don't have a very good reason to move. So there ought to be a continuum here where, yes, if you're reacting to an external stimulus like a baseball being thrown at you, then the influence of background noise is probably going to be very small or not at all. Whereas if the decision to act and in particular the precise time of that decision is up to you, is unconstrained, then it might play more of a role, right? And in particular in determining when the action is going to happen, not necessarily what action will happen. So in summary, this stochastic decision model accounts for the electrophysiological, behavioral, and subjective variables in Libet's task. It argues for a late commitment in spontaneous self-initiated movements, and it argues that the conscious intention has a meaningful and reasonably accurate relationship with the decision to move. And it also, by the way, suggests that a pre-event buildup might be a general phenomenon in nature rather than a specific mechanism in the brain. So we might look for this kind of pattern, for example, in stock market data or meteorological data. And mentioning meteorology here, I think this is one of the important future directions, and this is something that we're currently working on, is to move from an epoch kind of a model where we cut the data up into little segments that we analyze, and instead just looking at all the data, all of it, just like is done in weather forecasting, and try to build what I call a brain behavior forecast. So you're trying to take the data from now and in the recent past and forecast when and possibly also what movement is going to be made in the near future out to some time horizon. Now, if the difficulty in weather forecasting is any indicator, then this is not likely to work exceedingly well. We may not be able to predict very far in advance. If you consider the fact that a single human brain is arguably more complex than the entire Earth's atmosphere, and we still really can't predict the weather very well out past about 3 days in the future, then we may find equivalently in the human brain that we can't predict the choices or the onset of movement very far in advance. Thank you very much.
Thank you so much, Professor, for your valuable and thought-provoking presentation. It was truly fascinating to learn about the readiness potential and your research. We will now begin the Q&A session if we have time, a couple minutes, I hope.
Yeah.
Before we turn the audience, I would like to use my privilege as the moderator to ask the first question to kick off the discussion. You mentioned that BCIs often fail when they rely on the readiness potential, but drawing on your broader expertise, perhaps considering future technologies like advanced fMRI with AI techniques like LLMs, do you think it's theoretically possible to photograph or isolate a truly free will decision movement? I ask this because in criminal law, there are serious debates about neuroprediction and crime prevention. Putting the ethical concerns aside for the moment, technically speaking, do you believe we could ever reach a point where we detect a decision before a person commits a crime, for example?
Yeah, I think that's unlikely to work very well, no matter how sharp the data are that you have. Now, I could be wrong, but if you look at how long people have been working very, very tirelessly and with massive amounts of computing power to try and predict the weather, and still can't do that very far in advance very well. Um, I think it's unlikely that we'll be able to predict far in advance. So we might be able to predict close in advance, maybe a couple of few seconds. Um, I think given the complexity of the brain, even if you had very clean data and access to neural signals uh, at a very low level, um, I'm not convinced that that's gonna be very doable.
Thank you so much. Now we would love to hear from our participants. Please feel free to type your questions in the chat box, and we will try to answer as many as possible. You can also use the raise hand option if you would like to turn on your microphone and camera and ask the question yourself. Please do not hesitate.
Yeah, if you do have a question, just go right ahead.
Here we have some questions in chat box. Uh, Dora Su is asking, do the responses of evolutionary shaped neural motor complexes, encompassing both phylogenetically conserved primitive motor reflexes and subsequently acquired adaptive motor complexes, give rise to interdependent response patterns within a hierarchical domino-like framework of activation mediated exclusively through neural potentials and signal transmission?
Okay, that's a very packed question. So I think if what you mean uh, by hierarchical is that there are many factors that kind of layer and in determining the onset of the movement, if that's what you're thinking of, I do believe that there is a kind of a hierarchy, and I don't think that uh, background noise is the sole determinant of the initiation of movement in most contexts. So for example, if you do the task yourself or you ask subjects to just tell you, what were you thinking when you were doing this task? Of course, there are other layers. For example, people sometimes think, well, last time I waited kind of a long time before I moved, so maybe this time I shouldn't wait too long, right? So there are other kinds of preferences. We found a preference for the bottom of the clock. For example, when you're monitoring the clock, people tend to move a little bit more often when the dot is closer to the bottom of the clock. There can be other kinds of biases as well. So I do believe that there are layers and that some of them can be strategic that determine when you move. Um, but that in a task like Libet's, where there are very few constraints, then the time of the movement can, at least in part, be governed by noise.
Thanks you. Rana Çelebi is asking, if readiness potential arises from random neural fluctuations rather than intention, how might this help explain altered agency and control in psychiatric conditions such as OCD or impulsivity disorder?
Yeah, um, it might and it might not. Um, so those kinds of subjective feelings about of control and or lack of agency and control, um, those can also be uh, they can also have to do with timing, timing of the self-canceling features of your own neural signals. So We know that when you generate an action, your brain is already predicting the consequences of the action, the outcome of the action, already at the moment that that the brain activity is starting to bubble up that's going to initiate that action. And by your brain knowing precisely what's coming in the future, by knowing very precisely what's about to happen and the consequence of it, can cancel the effects of your own movement, which you want because you want to be able to tell the consequences that came from you from consequences that came from outside of you. And that relies on very precise timing. And if the timing is off, then the system might have trouble knowing, does that belong to me or is that coming from outside? And there you can have illusions of control and or lack of control and agency. So that can be for other kinds of reasons other than what I've been talking about.
Thanks a lot. From Zainab, I might have missed that part, but I was wondering how they explain crayfish having a readiness potential or premotor build-up without having a motor cortex. Does that tell us something important concerning our previous assumptions on the matter?
Yeah, I think so. So I think that what this is telling us is that there's a very general principle involved. The principle is this: when the determinants of an event involve a threshold and autocorrelation, when those two ingredients are present, then you're going to see an apparent slow buildup towards the time of the event, which corresponds to the threshold crossing, right? So even in a crayfish with a different kind of brain, as long as the system has those same properties, so there's a threshold and there's autocorrelation in the signals, then if you align to the threshold crossings, which we presume correspond to when the movement begins, then you will necessarily find a slow buildup beforehand, right? And just to be clear, in these experiments on the crayfish, what they're looking at are the transitions from rest to foraging, because a crayfish will just spend some time just resting, doing basically nothing. And then spontaneously at some point, it starts to forage for food. Then it will forage for a little while, and then it will spontaneously stop, and then wait and rest for a while, and then again spontaneously start. So it's the transition from rest to foraging. It's this behavioral state transition that shows that buildup beforehand.
From Kerim Özal, would you please handle and define the difference between spontaneous and non-spontaneous, which requires planning and intentional action, voluntary movements, in the course of neural antecedents?
Yeah, and this is something that's rather difficult to define very precisely, but I believe that there is something meaningful here. So you can say, well, nothing is spontaneous. Like even the case of the diving board, like the person jumping on off the diving board, it's not spontaneous. I mean, they know that they have to climb up the ladder, they have to walk out to the end of the board. Of course, they're going to jump off the diving board because this is a diving competition, and that's what they're going to do. So you could say it's not really spontaneous. It's just determined by all the things that came before and so on. But what we mean by spontaneous is that in the moment, in the immediate context, right, there is no immediate trigger to tell you specifically and precisely when to move. So maybe internally, that may or may not be spontaneous, but from a third person point of view, I think it's fair to call it spontaneous. And so that's what we mean by spontaneous in that context.
From Azra Melek Yaşar, does this model account for hyperkinetic disorders like chorea by assuming a lower decision threshold?
Um, I think perhaps it could. You could use the model to account for Huntington's, but there are other ways that you could account for Huntington's disease at all, because it's in that case Huntington's is it's more complicated than just having random movements at random times. The movements aren't like initiations; they are kind of meandering sort of ongoing movements rather than like moving and then not moving, right? Um, you you might come closer to this model if you were trying to account, for example, for Tourette's syndrome. That might be a little bit closer. And by the way, the Huntington's now they've recently, right? This was in the news. There's now a treatment for Huntington's, um, which is an amazing new development. Um, just as an aside.
Thank you. From Beyza Çetin, does stochastic accumulatory model imply that conscious perception, similar to motor initiation in limit-type tasks, emerges from random neural fluctuations reaching a threshold rather than from a discrete perceptual onset?
Um, I think the answer to that is yes and no. I think that sometimes in strange circumstances you might have this kind of thing happening, but in everyday life I don't think so, because I think that you have a a, a rich a context of visual and auditory and tactile stimuli uh, that are much stronger than any kind of small subthreshold background fluctuations. And that's driving the perceptual systems. Um, but it might explain the things that happen in these contexts where you develop visual, for example, visual hallucinations, which with like this what are called Ganzfeld uh, type experiments where you have a uniform, just colored background. You can't see anything visual except you just see red everywhere. And if you stay in that context for a long time, you start to hallucinate visually. And there, in that case, you might be able to explain that in terms of random threshold crossings.
Thank you. From Damla Durak, does conscious free will merely provide a time window to veto, stop an action that the brain has already initiated?
No, I don't think so. In fact, I think that was another thing that Libet just got wrong. Um, Now he claimed that we have free won't, that the reason we become conscious at 200 milliseconds before the movement is now we have this 200 millisecond window of time to stop the movement if we want to consciously, to intervene. The problem with that view is that we have very good data now from other labs, not from mine. Didn't do this work, but there's very good data that 200 milliseconds before you initiate the movement, that is in fact the point of no return. 200 milliseconds is the point where you can no longer prevent the movement from happening. By then it's too late. The movement's going to at least begin to happen. So if that's true, then 200 milliseconds can't be the veto window, right? It can't be the veto window and the point of no return at the same time. Those are completely at odds. They can't both be true. And I think that the data are pretty clear that it's actually the point of no return. In which case, no, you can't veto your action in the last 200 milliseconds before it happens. You can, however, cancel or interfere with an action at pretty much any time. And you can also make conscious decisions about the far future, like what you're going to do tomorrow or what you're going to do next week or next year. And those can be free decisions, right? Those can be acts of freedom of the will. So I don't think so. I don't think that that's the time window of veto.
Thank you so much. If we have all the questions, we can slowly end up this session. Thank you so much, Dr. Schurger, again. The
Oh, you're
perspective you've
welcome.
shared is truly valuable and really enjoyed listening you and meeting you. That is
Thank
an
you very
honor to
much.
hosting you on Cerrahpasa Neuroscience Talks by Cerrahpasa Nörobilim Kulübü. We thank you a lot.
Thank you for inviting me. It was a pleasure. Thank you very much.
Also, before leaving, can we take a photo?
Of course.
Okay, Rabia, if you can.
Okay.
Yeah, thank you so much.
Got it.
Thank you
Okay.
so much.
Thank you very much, and thank you for organizing this.
It was our pleasure.
That was a real
Okay.
pleasure. Thanks again.
You're welcome. Bye now.
Bye bye.